Welcome everyone to this special edition of the TCAP seminar series. I want to especially thank Ken Kephart for preparing this seminar for us. He's going to talk today about Augmented Lattice square design, and he's been working on some evaluation of spring barley. And so I know that we'll all benefit from his experience. Ken received both his bachelor's and master's degree here at Montana State, and then he went off to the University of Idaho to get his PhD. He's then served as a faculty member, as State Extension Small Grain Specialist at the University of Idaho, and then moved to the University of Missouri. Interestingly, he served as the first germplasm information curator for the wheat database of the USDA Grain Genes Project, which I didn't know, so I'm glad to know that. And he has been a member of the faculty here at MSU since 1998. He's a professor of agronomy in the Research Centers Department. He is a research agronomist, and he's superintendent of one of our research centers that is the Southern Research Center, and it's near Huntley, Montana, which is close to Billings. He's responsible for variety testing of all crops at this specific center and evaluation of germplasm, and he's a collaborator for the three small grain breeding projects. That's winter, spring wheat, and barley. And he also administrates the Southern Research Center. So we really appreciate Ken coming to share his expertise with the TCAP group, and we've already had a special seminar on a way to analyze
That's my
augmented
pleasure.
design, and so this is going to build upon
And I
that.
thank you for that introduction,
And
Jamie.
Ken,
I
just thank you so
appreciate that.
much for being willing
And
to
I
share your
also
time
understand that
and your
Jamie is going to
expertise
be
with
watching
us.
the chat screens
And
for questions. I'll be happy to stop and answer any question that might be needed in order to explain something in the seminar. But if you really want to have a more of a generic question involved, we'll try to address those towards the end. I'm sitting in my office in Huntley, Montana, in a very rural location here. I'm fortunate enough to have a T1 line into this office, so I'm able to communicate with you today. I've got my phone off, my door's locked. I will warn you that there is a situation that could develop. My office is only about 150 yards from the main railroad line for the Burlington Northern Santa Fe with a railroad crossing. And this microphone is supposed to be noise canceling, but if a coal train comes through, it could be challenged by the noise that's sometimes made by the Burlington Northern. So with that. When Jamie approached and asked me to talk about augmented lattice square designs, my first reaction is, why me? Although I was heavily trained in statistics in my PhD program, actually in both my master's and PhD program, I don't consider myself to be a theoretical statistician by any sense of means. Or there's a number of people out there that I'm sure that could address this topic from a theoretical standpoint much better than I can. But Jamie pointed out that I guess I'm one of the few, if not the only person that's involved in the triticide CAP program that's actually tried to conduct a study with an augmented lattice square design. And to be honest, I've only done that once in 2012. So I guess a lot of this is going to be a little bit of theory, and a lot of it's going to be based on the experiences that we have enjoyed in this past year in trying to make this work with our spring barley breeding program. There are some things today. Let's see if I can move on to the next slide here. There are some things that I'm not going to discuss today because I think they've been handled very well by other sources. In the introduction to augmented designs by Dr. Jennifer Kling, that's a presentation that she did earlier in the year and does a good job of providing some background information on augmented designs, how they're implemented, the characteristics of them, and basically why we are attracted to these designs. I've shown you the link in order to find the YouTube video from that presentation. I suggest that if you haven't seen that presentation, that you review it. It's very good in providing a lot of background information and how she's using augmented designs in her own breeding program. I'm going to be discussing an augmented lattice square today, and that is like most of our augmented designs. The augmented lattice square is a derivative or an extension of the basic lattice square experimental design. And if you're interested in looking for information from the standpoint of using that design or deploying that design, I provided some citations there. Lattice squares were developed in the 1930s. A lot of the work was conducted by Ronald Fisher and Frank Yates at Rothamsted. That work, they had a number of publications related to this, but it culminated in the 1940 paper that you see there, published in the Journal of Agricultural Science, simply titled Lattice Squares, and that's kind of the definitive hallmark of when these designs came about. Lattice squares are usually not found in very many statistical textbooks. But fortunately, Cochran and Cox devoted a chapter in the second edition of their book titled Experimental Designs. It's a very good discussion, providing background and some theoretical information, as well as some analysis work concerning the lattice squares. The last citation that you see there by Gomez and Gomez is in their book, Statistical Procedures for Agricultural Research. They actually, most of the discussion there is in that section listed in chapter 2, but they have additional discussion throughout the book. Probably it's not very strong in terms of discussing from a theoretical standpoint, but they do provide a very nice progression of step-by-step analysis for the data associated with this design. And so all three of those together would give anybody a good solid background in understanding lattice squares. I do have some resources listed concerning the specific discussion on augmented lattice squares, and all of that is been published by Walter Federer, who's noted as kind of the father of augmented designs. He proposed augmented lattice squares towards the tail end of his career, and has a number of publications. Like a lot of things that Dr. Federer published, some of these publications are missing little snippets of things that combined together. All three of those publications do a good job of describing the implementation of an augmented lattice square. The best single citation, of course, would be the one there published in the Biometrical Journal in 2002. That does a good job of explaining the construction, the analysis, the randomization procedure to deploy one of these designs. The last citation there was published back in 1983 by Dr. Lind and Puczynski. They were the individuals that developed the type 1 and type 2 modified augmented designs, and actually published a paper comparing performance of a replicated modified augmented design against balanced lattice square designs, and that's an interesting read as well if you're interested in using either a modified augmented design or comparison with a balanced, or wish to use a balanced lattice square design. So what are the attributes of a lattice square design? Well, quite simply, these are replicated single factor designs where the experimental units are arranged in rows and columns of equal size. And And except for the replicated part, you've probably heard of a Latin square, which is a single factor design with experimental units arranged in rows and columns. So this is an extension of the row and column designs that were developed. In fact, initially this was called a quasi-Latin square design before Frank Yates coined the term lattice square. But in the big difference is that this is a replicated or block design as compared to a lattice square, which is usually not a block design. When you read the literature, most often you see that the number of rows per replicate are given a K value. Well, since the number of rows is equal to the number of columns, that also describes the number of columns as K. And the number of experimental units per replicate, therefore, would be K by K or K square. That's pretty simple. Because each of the replicates, or what I will sometimes call whole blocks or complete blocks in this discussion, each of the replicates contain all treatments. This is a complete block design, and in fact, it's quite common to see in an analysis of a Latin square its comparison to a randomized complete block design from the standpoint of relative efficiency, because the same set of data can be analyzed as a randomized complete block design. And but randomization of the treatments is restricted by rows or columns, and so more correctly, this is a partially balanced incomplete block design, and this really leads to the power and strength of the design in providing some adjustment for spatial variability. I kind of have a diagram here. I hope this makes sense to you. In this particular example, I've set up what we would call experimental units or plots in the individual small squares here. We have 5 rows, 5 columns. So this group together, all the treatments that would be contained within this would represent the replicate or the complete block of the study. Now, we restrict randomization by how frequently a particular treatment or entry, in the case of the work where we're dealing with germplasm or varieties, individual genotypes would then be assigned to these blocks and would be restricted within these rows or within these columns. And because of that restriction, which I'll explain further here in just a moment, this is what lends itself to being an incomplete block, this block within the complete block here. Okay. Depending on how that restriction is imposed, you can have either, and of course the number of replications as well, you can have either what's called a balanced lattice square design or a partially balanced lattice square design. Now, in the definition of a balanced lattice square, the value K, remember this is what this number represents, the number of rows and the number of columns within each of the replicates, must be a prime number or the power of a prime number. And so in this particular case, if you're striving to develop a balanced lattice square design, K can be equal to 3, it can be equal to 4, it can be equal to 5, it can be equal to 7, it can be equal to 8, but it cannot be equal to the number 6. Obviously, the number 4 is 2 squared. K being 8 would be 2 cubed. But 6 is not a prime number, and it's not the power of a prime number. And so from the definition of a balanced lattice, you could not have 6 by 6 designs. There are 2 forms of this that can be implemented. The replication number can be k plus 1 divided by 2, and that's only if k is odd. So if, for instance, you have k is equal to 5, you would have 5 plus 1 is 6 divided by 2. You could develop what's called a balanced lattice square design with 3 replications. Now, in this, I say here, now I'll be honest, my tongue gets tied sometimes. I have sometimes trouble wrapping my head around how this should be said, and I'm trying to paraphrase from the text. But where the restriction here is where any pair of treatments, so in other words, any pair of cultivars or genotypes, can occur only once in the same row or in the same column. Perhaps a better way to say this, of all the possible pairing of treatments or genotypes, half of those will occur once within the set of rows, and half of them will occur within the set of columns. And based on the assumptions that we have for ANOVAs, basically that all treatments, in the case of genotypes, all treatments are, of course, represent a population with a common mean and a common variance. We would assume that the amount of error that's described by the pair treatments in the incomplete blocks based on rows and incomplete blocks based on columns would be the same. I understand from the literature that in application that's not always the same, and you have a choice of either ignoring that, which I don't know because I've never grown one of these or never used a balanced lattice design. But you have a choice here. You can either ignore that, or you can implement some of the methodologies or procedures that we use in partially balanced design, where you actually calculate different standard errors for pairs of treatments that occur either within the same incomplete block or a different incomplete block. The other kind of balanced design is where the replication number is k plus 1. It doesn't matter if k is odd or even. Again, it must be a prime number or the power of a prime number. I believe all possible combinations of those pairs occur, and they occur once, and only once in the same row and only once in the same column. And so this is, by definition, most publications that you see, textbooks, describing this as achieving treatment symmetry, so that you are measuring, in theory, the same level of error associated with incomplete blocks and rows and columns. And as a result, you actually only calculate one standard error for comparison to treatments in that form of the balanced lattice design. I'm not aware of anybody that's ever used a balanced lattice design, and I think it's pretty obvious that the amount of resources that would go into one of these are pretty horrendous. To grow, for instance, a 7x7 lattice, you would need 8 replications, and you could probably, most people would say, in the same amount of real estate, you could conduct 2 or 3 other different studies rather than just have one obligated to looking at 49 different genotypes. The other category are partially balanced lattice designs, and just about every lattice square experiment that I've conducted and those that I've seen conducted by other researchers fit into this category. There is no prime number restriction, or at least in theory, if there is a prime number restriction, it's largely ignored. And so you can have block sizes of 6x6, 10x10, 12x12, so forth, that don't fit the prime number definition. There's no restriction on replication number, as there would be in a balanced lattice design, and most frequently you see these replicated either 2 or 3 times. Common description of these, for instance, an experiment with 2 replications is often called a double lattice, and then those with 3 replications are called triple lattices. Any pair of treatments that occur only once in the same incomplete block, which can be rows or columns, but not both. So basically, in the restriction of the randomization, you're making sure that any pairs that occur only once. You will not have all possible pairing of treatment combinations in those incomplete blocks, and you're going to focus on the occurrence of those treatment pairs within either rows or columns, and then depending on which one you're tracking that on, you're basically going to ignore the other. And so there is, again, this is a design then where you would have uh, uh, in terms of separating means, you would calculate standard errors for treatments that do occur together within blocks versus those that do not. The analysis of a partially balanced design looks like this. Up there above is the model on which this is based, where whatever your dependent variable is based obviously on the population mean and is impacted by variation in reps, blocks within reps, as you can see here. And this, when we're talking about blocks within reps, we're talking about either the rows or the columns that are defined as the incomplete blocks within those reps. Genotypes, in this case, we're assuming this is a study where we're looking at varieties or germplasm. And then your interblock error would be the residual error, as you see down here at the end of the model down here. And so if you were to analyze a partially balanced design, you would see an analysis of variance laid out that looks something like this, where your source of variation is going to be reps in these, or complete blocks. That's what these represent here. The incomplete blocks, and those are incomplete blocks within the reps. You would analyze the treatments, the genotypes, unadjusted for any spatial variation in the experiment, and that's required because then your residual is what it makes up the interblock error in this analysis. Now, once that is done, components of the interblock and interblock error are then used to calculate a correction factor for the treatments. Once that adjustment is made, then treatment variability is then recalculated, and quite often you'll see both treatments unadjusted and treatments adjusted listed in the analysis of variance. And then, of course, a total here. So in a partially balanced lattice design with 3 replicates and k-value of 5, in other words, 5 by 5 blocks that we're growing here, you can see what the degrees of freedom, we wind up with 36 degrees of freedom, which would, in your interblock error, and then standard errors would be calculated as a weighted function of the intra and interblock error in this design. Fortunately, we have software that does all of that automatically. Proc Lattice and SAS is a very, very strong, very good product. I like to use it a lot in the analysis of lattice designs. The Proc Mixed and Proc GLM can both be coded and models set up to analyze partially balanced lattice designs. Of course, there's always, there's some suggestion that some of the adjustments made by Proc GLM may not be correct, and so Proc Mixed has become the preferred product for analyzing these designs, as suggested by SAS. Okay, so to summarize, both interblock and interblock errors captured means are adjusted for environmental heterogeneity. This is something I wanted to really drive home, is that unlike randomized complete blocks or many other designs that do not make that adjustment, this inherently makes an adjustment before we even look at other methods of adjusting the data. Inherently makes adjustments for some spatial variability. And the augmented lattice square design, being an extension of this, does exactly the same thing. One of the downside to lattice square designs, and this is based more on my own experience than seeing this written in text, but I have found that lattice square designs are intolerant of missing observations. And some of this makes sense in that, each incomplete block, because it's incomplete, is already missing, in theory, some observations, even though they exist in other incomplete blocks within the study. And so when you start removing observations and out of that, then the calculations needed uh, 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 simply are not, very quickly become unresolvable, I'll put it that way. The other problem that I also see associated with this is that one of the more popular designs that I see breeders use with lattice squares are the double lattice, where you have only 2 replicates. Obviously, if you have a missing observation of a genotype, you only have one other observation, and that's the very poor basis for trying to estimate any or use any sort of missing observation procedure to try to backfill that data set. So this is something that I kind of emphasize here, because lattice squares, they really work nicely if you have a complete data set. If you've got missing observations, the design does not work. But you do have a fallback in that, I mentioned earlier that every one of these designs can be analyzed as a randomized complete block design, and so whether you choose to try and backfill through a missing observation procedure, or maybe perhaps just remove that treatment and then analyze the data as a randomized complete block design, is up to the researcher. So why all this interest in lattice square designs? And a lot of that has to do with the degree of spatial variability that I, in particular, have to live with in this part of Montana, and in fact, this is not uncommon. It exists throughout Montana. I chose this picture because in the foreground you can see the vegetation that pretty much existed in this area at the time of Lewis and Clark, when they made their historic passage through the area. Sagebrush, a mixture of perennial and annual grasses and forbs and such. Where you see that sagebrush, this is probably in an area that has a glacial till soil. Everything you see from the greenery in the middle of the frame is man-made. And it's man-made from the standpoint that the Yellowstone River flows down through the middle of this picture with a very large irrigation district that was developed around the turn of the 20th century. And so you have an area, that flat area in the center there, that's been largely influenced by the flow of the Yellowstone and its tributaries since the last glacial age, with a lot of alluvial deposits up and down the river. And so, and because this production area is so narrow, in this picture you're looking at, I believe it's only 4 or 5 miles from the ridge that we're standing on to the one in the far edge of the frame. There's a lot of variation in soils, combined with things such as gravel lenses and sandbars and other things, and old creek bottoms that just when we try to use some of the more common designs, such as randomized complete blocks, just make it very, very difficult to discern differences in treatments. And what I've discovered is that if we're going to be conducting a variety trial, any chance that we get to deploy it as a lattice square, I will do so, because I have lots of personal experience where by doing so we go from a site that yields no discernible differences in entries to one that gives us a good separation of the cultivars that are being tested. And this is not common just to my program. It's also common among the 3 small grain breeding programs. Since we're talking about the barley breeding program at Montana State University, I worked with Tom Blake, our barley breeder, to develop this diagram showing the progression of material through his program from the initial cross until possibly a cultivar release. And one of the things you'll notice is that it's heavily leans on the use of lattices. Now, for instance, this preliminary yield trial this is the first time that we are looking at his material in any sort of replicated form in the field. That's a 16 by 16 double lattice, so each of the replicates contain 256 plots. Typically, those 256 plots are made up of anywhere from 13 to 15, 16, sometimes 18 check varieties, and then the remainder of those somewhere in the neighborhood of 235 up to almost 245 or so contain experimental lines that are either F7s, which are progressing immediately in the program, or some holdover F8s from the previous year. And again, this is a double lattice, so it only has 2 replications in the study. And then in almost every step of the way, this proceeds to an early yield trial. Most commonly, this is an 8 by 8 lattice with limited number of replications, 2 or 3. In our statewide testing program, the Interstate Trial, our common design in the barley breeding program is an 8 by 8 triple lattice. This is grown at 7 locations, and at least 5 or 6 of these locations actually grow two sets of the study, one under dryland, one under irrigation, and so we have usually 13, 14 or so experiments that are conducted in an 8 by 8 triple lattice, and an entry is usually in that study for 3 years. After 3 years, then we consider that for cultivar release, if it's a feed or hay type, if it's for malt, if it looks like it's going to proceed towards its use for malt, then there's additional processes that go on. But basically, you see lattice, you see lattice, you see lattice. So lattices have become the mainstay in our breeding programs because of their ability to adjust for spatial variation. I am one of the people that grow this preliminary yield trial, the 16 by 60 double lattice, and one of the issues that we face every year is how do we effectively harvest this without a loss of data? Because as I mentioned earlier, lattices are intolerant of missing observations, and it's been a challenge at times because harvesting 512 plots within a day is usually a pretty long, grueling day for my program. And so at the very least, we try to get through an entire replication of the study in one day and then follow that up the next day with the other. But we've had years where overnight something's happened, either a windstorm, hail, or something, and we may wind up not either harvesting that second replicate or have other issues that may arise that cause problems in the data. And so for a number of years, I've been trying to figure out what we could possibly use as an alternative to this 16 by 16 double lattice, something that would permit us to evaluate the same number of breeding lines, but would be more manageable from a planting and a breeding standpoint. And of course, that's what led me to my interest in the augmented lattice square design. Okay, this table is lifted directly out of the publication by Federer in 2002, and here this shows the partitioning of the degrees of freedom for K equals 3 to 11 augmented lattice square design. Source of variation you can see here, values of K. And so when you look down through here, you can see that some of these designs offer the opportunity to evaluate the number of new lines that are very, very similar to the number of lines that we've been evaluating in the double lattice, and in particular a value of k equals 7, or a 7 by 7 augmented lattice design. The other thing that this table helps point out is one of the things that Federer has proposed over the years is rather than measure interblock error simply as a function of the incomplete blocks nested within the reps, he has quite frequently suggested the use of what he calls a trend analysis, where orthogonal polynomials are used to assess the contribution of those incomplete blocks within replicates, either on a linear, quadratic, or cubic basis. And in a few minutes here, I'll show you how that is facilitated. But basically what this does is it allows you to partition out that source of interblock error into forms that are more likely to be significant. And of course, then you're not restricting your residual levels of degrees of freedom down to close to what would be a threshold mark in an augmented design. Based on those relationships I showed you in the first table, I developed this table to show you what the plot distribution is for an augmented lattice square. Again, now this is for k from 3 to 16 plots. In fact, I was amused in that Federer even suggested that you could easily go up to high as designs that where k equals 33. So 33 by 33 by 33, because k also represents the number of replication in these designs for a maximum design. You're talking about a study that's made up of 35,000 plots, and I'm more power to you if you want to go that far. But in my particular case, what I was looking for here was, of course, the total number of plots that were involved, and I show that here on a 7 by 7, with 2K, and you can have either 2K or 3K checks in the study. With 2K, which is the minimum amount of checks that you would need, because you need at least 2 check entries within each of those incomplete blocks in order to assess that interblock error. With 2K and a value of k equals 7, that means that we would have a room for about 245 experimental lines. So that matches up very nicely with the number of lines that we were assessing in our 16 by 16 double lattice. The design is very specific for the number of checks. So 2K is equal to 14. And 14 checks planted in 7 replicates is going to consume 98 of those plots. And that represents an occupancy rate, if you will, of checks at 29%. And this is one of the aspects of an augmented lattice square that some people might find unattractive, in that compared to other designs, it does tend to have a higher percentage of check plots, particularly on these smaller designs, higher percentage of check plots, which some people view as could be used to assess more experimental lines. That doesn't bother me so much. I like it. I like this approach in that I don't have to worry about whether I have enough checks or not. It's basically defined by the design itself, and so long as we satisfy that, if we need more experimental lines, we can always go to a higher value of k. If we have fewer lines that are than what are needed by 7 replicates, as opposed to going but more than what, say, a 6 by 6 would provide, we could just simply use fewer replications in the design, and it works quite well for that. Now, percent of check plots declines as you increase the size of the design. And as you can see, with 2K checks in a 16 by 6, you're only occupying 13% of the plots with check lines, and that would be comparable to some of the designs that are basically sold as needing fewer or less check varieties compared to, say, for instance, this augmented lattice. Of course, if you get up into bigger designs, you're consuming more real estate, and you might feel uncomfortable defining interblock error with only 2 checks within each of the incomplete blocks. And so Federer does provide the option of going with 3K and does provide methods for randomizing 3 checks within each of those incomplete blocks. And in my particular case, as you can, if you haven't picked up on this, I decided we were going to convert that preliminary yield trial into a 7 by 7 lattice with 7 replicates. Okay, so in 2012, the preliminary yield trial at Huntley used this design. So we 7 by 7 augmented lattice square with 2K or 14 checks, entries per replication, and this is as it was published by Federer. The 14 checks are arranged in a very systematic pattern, and I'll show you how that pattern is developed here in just a moment. Remaining plots were randomly assigned to 245 unreplicated F7 and F8 lines. And then grain yield, and I made a mistake when I was preparing for this presentation. When I was doing my final set of slides, I, by mistake, told my ProcMix to access the wrong column of data. We're still dealing with grain yield, but it's in bushels per acre rather than kilograms per hectare, but that does not affect the interpretation of the data at the end of the day. So we'll just go ahead and continue with that. I analyzed the data using ProcMix, and I did so in 3 different ways here. One was data was left unadjusted. We're not going to adjust based on likelihood. So we're just going to use a type 3 sums of squares approach for that. And actually, ProcMix worked quite well for this approach. You don't have to use ProcGLM. We're also going to analyze the data adjusted using REML to provide what are known as best linear unbiased estimates for the unreplicated lines, and then data adjusted by REML to provide best linear unbiased predictors for the unreplicated lines. And obviously in this particular case, I'm defining where we're going to be calculating estimates. We're defining unreplicated lines as fixed effects, where we are calculating predictors, unbiased predictors. We're assigning unreplicated lines as a random effect. So how do we go about assigning checks into the rows and columns of this design? So here we have a 7 by 7 lattice. This is a single replication. My numbering scheme usually is in the usually rep 1 is in the hundreds. Rep 2 would be a 200 series set of numbers. Third rep would be 300 and so forth. In Federer's paper, the assignment of checks is based on 2 diagonals that are established within the initial block. And so in rep 1 here, we've actually assigned checks 1 through 7 to 1 diagonal, and in the example that was used in his paper, he assigned that to the diagonal that just cuts straight across from corner to corner in the replicate or the complete block. The second diagonal is then moved over 1 to the right. So these 6 cells then, check 8 is continued through check 13, and then the complementary plot that fills out the seventh plot in that diagonal is down here in plot 107, where C14, the 14th check. Now you have a choice here in that one of these diagonals will remain static. The checks will appear in the same location, relatively speaking, within each of these blocks from rep to rep to rep. Therefore, they stay static where they appear within the incomplete block from rep to rep to rep. The other diagonal, and in his example, he uses the second diagonal. This is a dynamic diagonal in that you are going to move or reposition these checks in order to change their order and where they appear within the block, in the replicate, excuse me. And so I have a diagram here that shows this. So in this particular case, you to develop the second block, the second replicate, or the second whole block in this study, we would take this check number 8, and then we would move it down to plot 116, which would push check number 9 down into 124, check 10 down into 132, so forth and so on, all the way down until you get down to the bottom of the stack down here, in which case then C13 is going to be relocated back over to plot 107, and then C14 moves back to the top in the diagram occupying plot 108. And so what we've done is we've changed the pairing of the checks in each of those incomplete blocks, and that pair does not exist as far as the incomplete blocks in either, excuse me, does not in any of the incomplete blocks in the first rep. And then of course you would reassign plot numbers to signify that it was in fact what we're doing. Sorry, too heavy-handed here. So here, this would be the result of moving the plots within that dynamic diagonal, if you will. And so we would continue this process until we filled out all 7 replicates, and you wind up with a default arrangement that looks like this. Now I don't show you what the individual checks are. You'll have to trust me on that. I've just identified the plots where the check values occur. But this arrangement does have a symmetry associated with it, in that because you're not pairing a check that exists within a diagonal with any other check within that same diagonal, and you're only pairing the checks between the 2 diagonals, then each of these incomplete blocks, whether it's rows or columns, is going to have a pair that happens once and only once, basically for half of the combination of pairs that could possibly exist among all those checks. And so therefore, if you are forced to remove a particular replicate out of the design, or you wish to plant fewer replicates, yes, you're removing a set of pairwise combinations but you're also removing in rows, but you're also removing a complementary pair that would also exist within those columns, and so you maintain the symmetry that you started with throughout the study. Now you don't have to do that this way. You don't have to use this default arrangement, although this is what we wound up with deploying in 2012. There are alternatives that you can do, because I realize some people don't like the concentration of blocks in one particular zone, say in the middle of a replicate. And so there are other ways that you can use the same sort of system and wind up with a slightly different arrangement. And in this particular case, what we've done here is the first diagonal, the one that originally started in plot 101, is now starting in plot 103. And so we would continue down that diagonal until we get down to plot 135, and then the plots that finish out that diagonal within this replicate would be plots 136 and 134. And this is the static diagonal as before, and then the dynamic diagonal again starts in 108, goes down to 148, and then the last one is over here in plot 107, and that's the one that we will continue to cycle through as we did before. But by moving this first diagonal over or down 2 plots, then we open up an area where we have unreplicated lines that are dispersed in between checks in a little bit more open fashion than what we're accustomed to. And the other option that you have is that once you've assigned the pairs within a replicate, nothing stops you from taking these blocks and either swinging them left to right or flipping them top to bottom. And so in this particular case, I took rep 2 and I swapped it top to bottom. And so check C13 moves from the bottom of this edge up to the top over here, and so forth. The pairwise combinations do not change within each of the incomplete blocks. And so by doing it this way, you can further mix up the assignment to make it look more randomized. And in fact, if you do this, you can wind up with something that looks like this, so what I call an alternative arrangement, where in this particular case, we started out, again, I moved the 2 diagonals apart by 2 plots, and then I believe I flipped replicate number 2 and swapped left to right replicate number 6. And you can see that, makes a very nice and pretty pattern. I mean, you could sew quilts that look this nice from this design here. And but you get the idea is that what we're doing is we're decreasing the mean number of experimental units that are distant from an unreplicated line to a check in this arrangement versus what we had in the default arrangement down here. So with that, let's talk about the design that we deployed in 2012 and some of the results. Now I'm going to start out here talking about how we went about analyzing this. I'm not going to show you the total, all of the code that we used with SAS, and if you haven't picked up on it by now, I'm a SAS user. I'm a very strong proponent of using SAS. I'm not going to show you all of the code that's associated with my analysis. What I'm going to show you here is basically the framework, what we set up in each of the proc mix statements to make this analysis happen. So here is the core of each of the proc mix statements that I show you. If you're not familiar with SAS, the data equal statement says we're going to analyze the data that's contained in this temporary SAS data set called AUG-LSD7. In this particular case, we're not going to make any adjustment based on likelihood methods. Instead, we're going to analyze this with type 3 sums of squares, and so we can force that proc mix just to work with type 3 sums of squares by using that statement. Cove test is I'm asking for a summary of the covariance matrix in the analysis and some of the calculations associated with that matrix there. Class statement defines our independent variables. So in this data set, AUG-LSD7, We have columns that identify reps. We have columns that identify row and column, and those are integers in both cases that go from 1 to 7. We have a column that identifies the check varieties, and those are identified by an entry number. And then the unreplicated lines, actually this is entry, this is actually all of the lines, both unreplicated and replicated in the study. And I'll show you here in a second how just how we sort out the unreplicated lines from that entire list. Model statement, yield. In this particular case with proc mixed, we only give it the fixed effects in the model statement. So we're saying checks, entries are fixed. And then I have a covariate, if you will, in the data set called new. And that is either the number 1 or the number 0. If it's in a record that contains a unreplicated line, that is the number 1. If it is a check value, it is 0. And by using this approach, proc mix will only look at those entries as a source of variation that are unreplicated lines. Okay, that's the core in this step of the analysis. Followed by, if you have random effects in a mixed model, and which of course is we do, in proc mixed you have to list those separately. And so I actually will use one of 4 different random statements here. I mentioned earlier that that a lattice square can be analyzed as a randomized complete block design. This is kind of cool in that one of the frustrations in dealing with augmented designs and trying to assess whether or not they're functioning as promised is trying to make some sort of comparison to other designs, and that's very, very difficult to do because you can't grow or utilize a different design in that same exact spot in the same year that you've conducted the study with whatever your first design is that you're looking at. And so what people tend to do is just make a comparison of relative efficiencies of that one design with unadjusted versus adjusted data. In the case of a lattice design, we have some options here, and in this particular case, I'm going to be analyzing this as a randomized complete block design. So I'm really only including rep in the random statements. Because of the symmetry we have with the pairing of the checks, as indicated by Federer, I have a choice here. I can analyze the data with rows as incomplete blocks. And so that's what this statement will do. I can analyze it with columns as incomplete blocks. And that is what this statement will do. And then earlier I mentioned that we can also have the option of using a trend analysis with orthogonal covariates. And so these are R1 and C2 or orthogonal covariates that are in the original data set. I won't go into how we code those into the set because Federer has used this approach quite exhaustively in a number of different designs, and that's well published. But basically, we're using orthogonal polynomials as covariates to look for, in this case, linear effects of rows as incomplete blocks within reps, linear effects of column, the interaction of those two, the quadratic effects of rows within reps, the quadratic effect of columns, the interaction of those quadratic row and column effects, and then the cubic effects of rows and the cubic effects of columns. And actually, if you had a study large enough, you could probably keep going with even more complicated orthogonal polynomial relationships. But in this particular case, you're going to run out of degrees of freedom real fast. And so this is kind of the limit as far as we can go with a 7x7 design. These are generated in SAS. We use proc IML, and that code, as I said, is out in publications on the internet. And so basically here we have for our rows, we define our rows. I mentioned those are integers from numbers 1 to 7. And then we ask IML to produce a set of orthogonal polynomials for linear, quadratic, and cubic. And so basically what you're doing here is you're recoding your incomplete blocks with orthogonal polynomials that represent single degree of freedom comparisons, where all of these would add up to 0, and then the cross product of all of these would then also add up to 0. So it's kind of a neat way to do that. And you repeat that process for columns, and then through a series of merge statements, because you've already coded the data with row and column numbers, you simply use a series of merge statements to apply this data to your original data set. So let's look at some relative efficiencies. And here, what I'm trying to do is compare. These are 4 unadjusted models, so we're not looking at any covariates other than in the trend analysis on the interblock error. We're using type 3 sums of squares, and in this particular case, the unreplicated lines are considered fixed. And as you can see, by using rows as blocks, the relative efficiency compared to a randomized complete block only increased by 10%. And I was a bit surprised at that, because quite often when I grow a lattice in a full in one of the other experiments, it's when we say when the lattice kicks in, it's not unusual for us to see numbers that are at least 20-25% above what you would have in terms of efficiency for a randomized complete block. If we can look at how well columns as blocks did, there you can see a big improvement in relative efficiency, basically 249% improvement over the RCBD. And then in the trend analysis, I was a bit disappointed in this. I thought that actually we would have the highest relative efficiency with the trend analysis using the full complement of orthogonal polynomials, that we would have our highest relative efficiency. And so I was a bit disappointed in this. And in fact, compared to columns as block, you can see it's only 6/10 the efficient as the best analysis that we have on here. So I got to looking. I decided what I would do is I would go back in and on this trend analysis, look at the partitioning of those sources and see if there wasn't something funny going on. And on the face of it, when I first looked, I was looking at type 3 sums of squares. And excuse me here, I have an issue here. Okay, well, I was looking at this analysis, and I didn't see, obviously nothing struck me as being odd about the partitioning the sums of squares, the mean square values or what, but then I noticed that virtually every one of these was not significant over here, which I found hard to believe given the influence that an overall assessment of columns within blocks produced, that at least some or a combination of the column orthogonals would be highly significant. And then I noticed that there's something going on here because our degrees of freedom are not adding up. Each of these should be 7 degrees of freedom, and instead they're 6. We're only showing 10 degrees of freedom for the check varieties, and that should be 13. And then there should be 244 degrees of freedom for the entries, and that also does not add up. So what I decided to do then, and the reason why I asked for that covariant test was so that I could look at the parameter estimates. And in this particular case, the estimate is actually goes through, if you've ever done estimated or expected mean squares, calculated those, it shows you two things. One is it shows you which would be the correct denominator to use in an F test to partition out to see if you're testing for the effect of that particular parameter is significant or not. But also it permits you to actually calculate what the specific variance is associated with that. And that's exactly what ProcMix has done here. And you can see here that we have a number of these that are actually negative in this analysis. And so if we assume, as quite often you see in the literature, it seems to be the preferred method, is you assume that these are 0, and so in this particular case I'm going to remove these and then rerun the data in what I would call a reduced model. We'll see what the impact of that would be. As it turns out, I had to run this through 2 iterations by removing these 3 here, the linear effective rep, the combination of linear effective rep and columns, rows and columns, and the quadratic effect of columns. Turned out that I believe the quadratic effect of rep was also, I believe, turned out to be a negative variance, so I actually wound up removing that one as well. And when I do that with a reduced model, you can see that the relative efficiencies come up in, and although still not as great as columns within blocks, it is a substantial improvement in adjustment. And so, and as it turns out, most of the components that remained in this reduced model were some factor associated with columns within the reps for our interblock error. And then upon further look at that, I decided we would take out all of those coefficients that were deemed not significant in the test. And so we wound up with just actually one component, which was the linear effect of columns within rep, and resulted in this relative efficiency here, which is not a, numerically it's less, but basically we don't see any change or from the reduced model. So we're not, I guess what I'm saying is we're not penalized by either going with the reduced model or the significance, the single significant factor there. Okay, so taking this analysis, I also wanted to look at how we were doing in terms of discerning differences in our fixed effects. And so here you can see the probabilities of a greater F for check entries and for unreplicated. And what surprised me, even with all of the adjustment, we were not still discerning a difference among the observations of the unreplicated entries with the columns as a block, even though that adjustment was nearly 250% compared to a randomized complete block design, whereas the trend analysis, both of these resulted in probability levels that would be considered significant. And of course, as always, we would want to look at the residuals, and SAS provides a mechanism. These are graphics produced by the ODS system where you can look at distribution of the residuals and distribution, I guess, against the predicted, and then the distribution of the residuals as a percent. And the histogram over here, which is pretty nice. So in the next step, we changed our method to REML, and we left unreplicated entries as fixed effects, and then ran through the 3 different models again. 4 different models, excuse me. And one of the things I picked up on immediately in the trend analysis, that as you remember in the type 3 sums of squares, ProcMix reports negative variance terms. In fact, when you go to an adjustment based on probabilities or likelihoods, ProcMix says, well, negative variances don't make sense, and so it automatically zeroes those out. Again, that could be appropriate or inappropriate. I don't think we're penalized by this approach in this particular experiment. But one of the things you must be aware of, even though it turns these to 0, you better be watching your denominator or your error degrees of freedom, because it still partitions out the degrees of freedom from these effects from the model, and so you're being penalized down here in making a valid comparison for your fixed effects. So in other words, you have to literally take those out of the model. So in this particular slide, rather than showing you the full trend, I'm just going to show you the reduced and the significant, and look at the adjusted models using REML. And as you can see, the numbers basically have not changed. Most of our adjustment in relative efficiencies are inherent to the design of the lattice square, rather than based on adjustments made by REML. But there are some changes in the probability of F when you look at the test for the fixed effects on the checks and the unreplicated entries.
In the chat,
columns and blocks
I'm not
now
sure you can't
do
see
show
it,
a significant
do you have a
difference
software
in
or any
unreplicated
program that
entries. The reduced model
helps
trend does
in
not.
assigning checks and test
And
genotypes to
the single. So
plots?
basically, this is telling me that most of what we're seeing here in the columns as block is what we're picking up on in that linear column effect within replicates over here. So, and then lastly, we're going to be looking at REML with the data set. Only in this case, we're no longer going to assume that entries are fixed effects. Instead, we're going to assign them into the random portion of the model and run that. And I believe in this particular case, I only show the significant trend analysis with the single orthogonal polynomial used in the set. And again, we don't see a big change in relative efficiencies as we go from calculating the estimates to the predictors. I will point out, all of a sudden now rows, it shows you that basically rows aren't measuring hardly anything out there because they virtually have the same relative efficiency as a randomized complete block design. And then down here, we're looking at comparisons, in which case both columns as block and trend show, in this case with columns as blocks, unreplicated entries are highly significant as well. Not much changes in the check entries. I'm not surprised at that. And then the trend analysis. So at the end of the day, when we were generating data for Tom to look at in the breeding program, I chose the model where columns serve as our incomplete blocks in that final analysis.
Are there any other questions?
And as always, we want to look at the
people maybe are
residuals,
thinking,
and this is
Ken, I really want to thank you for all of this information.
RCBD design,
and I noticed that some
effects,
people had to drop out,
and
probably because they had another appointment.
The distribution of residuals against predicted mean.
No.
And you can see we have some issues here.
I just want you to know that it's
been
recorded, and so people can come back to it. And so I'm glad you took your time and went slowly through it, because
distribution
then it'll be very clear for people to come back.
Actually looks pretty good, but
to come back to it.
The
so that's why
data
I didn't push you on. I wanted it to be clear.
Is from minus 21
but there is
to
someone
16,
typing in the chat box, so we'll see what question
here.
they have.
When we analyze the data with columns as blocks, which I previously said I thought was the best fitting model, the appearance of our residuals appear much more random, and we do attain symmetry on our residuals, as the spread is now from about -11 to a little less than -12. And so
just so everybody,
that,
people are still typing, just so
in terms
everybody
of
knows,
using, if we
we
were
will
to use
post
this
this as soon
data,
as
say, for
possible so you
heritability
can come
analysis,
back to it.
this would be a much better fit.
Because I think there's a lot of information there
would be
that,
less
that people would probably like to refer
biased,
back to.
I guess is the best term. Should be unbiased, but it's at least less biased than
Schiffer,
any of the
I
other
noticed, did you have a
models
question,
that we
or?
have run here so far. And
There
then
it
the
is.
last step, of course,
Perfect.
is if you are a breeder, you would be interested
Go ahead, Ken. Can you see
what
that
sort
question?
of changes would happen as you go from fixed and random effects. And so in this particular case, I ran the two column as block models, either with lines being fixed or random, and then ran Spearman's rank correlation coefficients, and you can see there's pretty much there's highly significant general agreement in the rank order of the lines, whether they're random or fixed. But obviously, the shrinkage factor associated with analyzing these as a random effect, it would be more desirable for genetic analysis. And when I look at it and I rank these, now I'm not going to show you the whole data because there's pages and pages of data, but these are the residuals, if you will. So these would be estimates for the fixed effects. These are predictors under the random effect. And I show the ranking of each of those. And if I was a breeder, say, for instance, if I wanted to have, say, select the top 10% of these lines, I would rank them. In this particular case, I've ranked them by the top 25 fixed lines, and then look for differences between the fixed ranking and the randomization, or excuse me, the random ranking. And as you can see, and as a random effect, there are 4 lines that I would in fact consider that I wouldn't normally consider if I was just analyzing this as a fixed effect. And so not a large number, but could be significant. Well, I thank you very much, and I hope that you've understood what I've tried to share with you today. I guess on that note, I'll open it up and see if there's any questions. The chat window is empty. I'm surprised. Okay. Oh, okay. Okay. Basically, what I've been using is using Excel with setting up a sequence of randomizations. This is not very complicated here. First of all, the assignment of checks.
Well, I
you
think
saw
more
that was
people are
very
dropping out.
systematic.
There was
and the assignment,
Silvano
we have
is got
14
another question
checks
for you, but I think this will be our last
in this
question,
particular
and I
design.
want to
And
thank
so what we can
everybody
do
for
is we can, in
attending.
Excel, I can list
Here's
those 14
Silvano's
checks, and then
question. How is
in the
spatial
next column, I
variability
create a
taken
dummy
care of?
variable that's based on the random function.
Go ahead.
In Excel, and basically I sort based on that random generation function, and then the order which those checks show up is the order which I assign them to check number 1 through check number 14, and that's pretty simple because the systematic pattern of the checks within in the study is already defined by the design itself. Then to assign the unreplicated lines, I basically do the same thing. I list them in a spreadsheet in a column, and next to it again I have a dummy factor that I list that's a function of the randomization function in Excel, and then sort that from top to bottom, and that's the order that I plug them into those individual plots, starting with plot. I guess we go from 101 out to possibly 749, and so those are assigned in that order there, and that And then once I do that, my assignment of plot numbers is automatic, and that's how I wind up with this design. It takes about 5-10 minutes for me to go through that process.
Great. Ken, again, thank you so much for being our resident expert today. And we really appreciate it. And we have a discussion post opened on the Plant Breeding Training Network.
I ran long, and
If there are
been
further questions or
notification,
discussions, shoot a question that way, and we'll try to get it to an appropriate person that can answer it. And thanks everybody for attending.